Hello, everyone, and thank you for joining us for this presentation all about neonatal donation. This presentation is scheduled for roughly 60 minutes, including time at the end for Q&A. You can enter any questions you have in the questions section of the GoToWebinar control panel, and you can also find a copy of today's presentation slides in the handout section of that control panel. Joining me today is Gina Dunsmith, Laura Le Guin, and Dr. Gloria Pryhuber. To get things started, I'm going to pass things over to Gina.
Thank you, John. And thank you for the audience today for taking the time to learn more about this very special area of donation through neonates that are being referred. So I'm going to start back at our first story, which started with a family, Bethany and Eric Kunkel, who found out early in their pregnancy that their son, Amalia, was diagnosed with anencephaly. For those that do not know what anencephaly is, this is a diagnosis where the skull does not develop and a large part of the brain also does not develop. However, the organs are pristine. One would not know this without the experience that we have had. But despite that, back in 2012, there was really very little that was being offered to families that were exploring donation. And like the Konkels, they pursued many, many avenues and spent the next 6 months of their pregnancy being rejected and frustrated by the attempt to try to see what they could do. We were finally contacted by the Life Connection of Ohio, who referred the Konkels over to IIAM. And we had at the time 2 researchers that were interested in the liver and pancreas that were from very young ages of donors. And so we were able to kind of walk through this, the blind leading the blind, on this type of a research exclusive donor. And where that will come into play is that you can see here that there's very limited opportunities for donation when it comes to transplantation. And typically those are limited to kidneys or heart for valves, both of which are age and weight dependent. If you think about an anencephalic, the heaviest part of a baby when they're born will be the skull or the head. And with the skull not being developed, it certainly compromises the weight of the baby. And now donor families that where a diagnosis of anencephaly was received are trying to challenge the world of donation to see if their baby, if the weight would be adjusted and to have their baby still be a candidate for heart valve. But as that's moving forward, we in the world of medical research have been moving forward with an expanding network of researchers, and we'll talk a little bit about what we're able to do. But the most exciting part of what we do with these families is enable them to realize that the donation of the organs and tissues from their baby will also go on to impact other babies and children and ailments that they will live with or die from in their lifetime. So ultimately, through donation, be it transplant or research, this is families that really want to make a profound difference for their child. Our experience has led us to always working through the local OPO, wherever the referral comes from, whether it's the hospital, the family, or the OPO. We route everything through these subject matter experts in the areas of authorization, the donor risk assessment, serology draw, and the organ recovery skills. There is no information that really can be obtained from the baby. So much of what's happening is from the donor mom, which does include the blood draw and the DRE. Early referrals are very, very key in our success in being able to do this, largely because we want to hear about the diagnosis and we want to learn about how that diagnosis is going to impact organs or tissues once we are seeing the baby being born or through the progress of ultrasounds and what that may reveal. You'll hear about families making a birth plan, and where that's relevant is that they may reserve time immediately after birth and after time of death to be with their baby, but understanding that that may preclude opportunities for donation as we together explain what the path is that we would like to take. And so we just honor what that family's decision will be. Having the right relationships in play, which include the staff from the OPO, the hospital, the donor family, the labor and delivery units, are key also to making sure that we can walk down a very feasible path. As I said, we have a growing network of researchers, and to date, we are working with about 17 different research studies. Not every call that we get is a donation referral that we can move forward with, and sometimes it's prior to the donation happening, and sometimes it's in the middle. So a diagnosis, as I said, may preclude the organs from being viable. So our experience might be just a couple of examples with trisomies or limb-body wall syndrome. This is where the organs can be significantly compromised. There may be low amniotic fluid, which again would deprive the nutrients and the baby from advancing into viable organs and cells. There are circumstances, and the key is from time of birth to time of death, and then from time of death to time of recovery, where the warm ischemic interval will be, again, challenged. And so, again, the key is communicating with that donor family and with the OPO staff, who is the contact to the donor family, and ensuring them that the if-then paradigms are all being explained so that regardless of whether their birth plan or whether their circumstances from time of death to time of recovery are going to impede the ability for the organs to remain viable. Logistically, also, that might come into play where, in fact, the donor recovery area may be different from where the delivery was. And so those factors have to come into play. Again, communicating and making sure that everything is laid out and so that we know what we're facing. To date, we have done 110 donors, and from those donors, we placed 381 organs and tissues. Primarily, these are main organs, with the largest amount, 80% of donors, being able to donate lungs. And we've also now included reproductive tissue, skin, thymus, any of the organs which a research entity may be looking to explore the cells. And again, that is where the viability, that warm ischemic interval is going to come into play. Those organs and tissues, it's key that they remain viable so that the researchers who are taking these cells are able to look at the viability and understand the role of those cells. Nearly every one of our donors has been a multi-organ donor. And if at times, if there is a single donor or, a single organ or en bloc lungs, where the degree or the outcome of the organ placement may not be what the OPO would like to support or what the family had in mind, the bottom line is if the OPO and or the family are willing to move forward, then we move forward with you to make sure that the intention for donation can come to fruition. Through our experience since 2012, this is information that we have gathered that I think could be of interest to the listening audience today. If you look from birth to time of death, the two categories that are the most common are that the time of death will happen within the first 1 to 6 hours after birth. And then there's as many donors that have actually survived more than 24 hours. But I can tell you it's very uncommon that they will last more than 36 hours, and typically time of death will happen shortly thereafter. The cause of death has primarily been a diagnosis of anencephaly, with the remaining diagnoses being an HIE or some other anoxic event where the pregnancy was otherwise normal. And then the gestational age, we have sections here where, according to the medical definitions, you'll see that preterm is from 20 weeks until 36 weeks and 6 days, being one common area. We have early term, which is 37 weeks to 38 weeks and 6 days, and then full term, which is 39 weeks and 40 weeks and 6 days. But in the preterm, we have rarely, if ever, had a baby that is at the younger aspect of the preterm definition. Typically, the donors, if it's in this pocket here, are at 35 to 36 weeks of gestational age. And because of medical risk to baby and or mom, there has been a medical directive to schedule an inducement or a C-section to make sure that the baby has the best opportunity to survive, or to make sure that the mom is protected from whatever may be going on with her. The most important slide that I can leave with you today is that 83% of all of our donors, 91 of the 110, were research-exclusive donors. So without an opportunity to donate for medical research and medical advancement, these families would not have had that opportunity to leave a lasting legacy for their loved one. And despite the fact that many families absolutely are praying for a miracle, where the diagnosis may not be correct, where the diagnosis and the condition upon birth will still allow that baby to thrive, they do understand that miracles work in many different ways, which may include the legacy that this baby will leave for other children and their family. Much of the research, again, is babies helping babies, and whether that's through the significant amount of work being done in the area of type 1 diabetes, where through stem cell isolation or beta cells, that researchers are trying to determine ways to regenerate these cells, to replace the cells that patients' bodies destroy, or to find a way to prevent or reverse type 1 diabetes. And then other areas of regenerative medicine that many of the other organs are impacting do allow, again, for regenerative properties to be explored with the cells. And the diseases are typically impacting chronic airway disorders or kidney disease, heart disease, and intestinal disorders, some of which you'll hear in detail today with the researcher that IAM has been working with for quite some time. We've made some great strides since our first donor, where we were very blind and there was not much known, certainly not much talked about, where now the media has really embraced these types of donations, whether it's a local news story, as you see on the left, with a baby from South Carolina through Sharing Hope, or if it's a baby on the right whose story is highlighted in January People magazine. One of the other things that IIAM has been able to do is that in December of 2018, we launched our Galaxy of Guardians program. And this is a package that's being presented through the OPOs and then presented to the donor families to allow the donor family to name a star after their loved one, so that at any time that they desire, they can look up and see that their baby's light is still shining upon them. It's not uncommon that we may have a researcher that does write back to IIAM or send another email that explains any additional work or the outcome of the work that they've been able to do as a result of the donation. And in many of the parents' perspectives, this is their opportunity to continue to parent their child, because these are new milestones that they enjoy hearing about. I said in the beginning that there was very little information that was available. So as we have been going through this process, IIAM and our parent company, MTF, has collected a group that we've created a steering committee that were bioethicists, neonatologists, nurses, reproductive lawyers, donor family representatives, and OPO executives. And together we have put a paper together that describes the current perspective from an ethical, medical, legal, and operational manner, and that allows organizations like IIAM to determine how it's best to proceed with a referral of a neonate, especially if it's a preterm inducement, a preterm delivery. And I can tell you that the one underlying requirement is at a time of birth, no matter when the birth is happening, that if inducement or early term delivery happens, that the desire is to have a live birth. And finally, if you are interested in supporting this type of donation, if you look at the IIAM website or even your local OPO, there is so much that now has been developed. These are resources that help the staff, help families, help communities, help the donor and hospital professionals move forward, how to answer the questions. There are other references that are available. I think Laura Le Guin from Louisiana Organ Procurement Agency will share what LOPA has done for their families, since they've had so many donors to date. And I will now hand the slides over to Laura, and she can tell their story. Thank you.
Thank you, Gina. All right, guys, so I'm going to kind of go through my presentation to give you from an OPO perspective. So we started our program in 2013, and to date we have done 12 successful donors. With each case, we've learned something new and valuable. And if there's anything you'll take away from this presentation, just know that each case is unique. Every one of them is going to be different in some sort of fashion. As our milestone, we were the first OPO to recover neonatal intestines. We were the first OPO to recover neonatal ovaries. And we were the first OPO to have a flag raising ceremony. This slide here is a picture of everyone that came out for that flag raising ceremony. So when I tell you that each case is unique, it is unique from the diagnosis, the family expectations, as well as the hospital staff. Now, some of the diagnoses that we've worked with have been anencephaly and Potter syndrome. I would encourage you to familiarize yourself with this diagnosis and familiarize your staff with the diagnosis. Be comfortable with the appearance of the baby. Know how these diagnoses might affect mom, so that if mom were to go into labor early and she calls you and says, or she calls and says that her amniotic fluid's really low, that you would know why. And it's important to develop a wish list or a birth plan with your family. This came after doing our first case, is when we realized that these moms just have so much that they want to do. And like I said, each family is different. So developing a wish list can make things run a lot smoother on their special day. A lot of families like to have a photographer. Spiritual needs. Some families want the baby baptized, or whatever religion they may be. Some may only want certain family members allowed to see the baby. How much time do they want to have with the baby? And are there any special things that they want to do with the baby? Um, like I said, every case has been different. It's been from the mom who wanted immediate skin to skin contact and for the baby to never leave her room, to the mom that did not even want to see the baby. So it's important to do this early with your family, find out what their wishes are, and meet with the staff of the hospital and let them know the wishes so we can all make this happen. This family here, this is baby James. He had Potter syndrome. So we had this birth plan ready to go, and mom went into labor early. She had a photographer, had the phone number. The photographer was out of town. So this uh, taught me that we now need a backup plan, for when this kind of stuff happens. We were able to find a photographer. She was great. She came in, and I learned so much about how we can get in touch with people on the spot when we need be. Now, after recovery, something to consider putting on your wish list is the family going to want, baby after recovery? Some families do, some families don't. Another thing to consider is moving the mom to a more isolated room. You hate for mom to be in the middle of the hallway where she can hear other crying babies or other moms laboring. Um, Also, beforehand, know what their funeral home arrangements may be. If they have a funeral home picked out, if that's something that you at your OPO needs to arrange for them. Again, just the pre planning for these is critical. During one of our wish lists, for the first and only time, we had a family that wanted to meet the recovery team. And this is baby Clark. Baby Clark had anencephaly, and Clark's mom on her wish list was to meet the recovery team. I did not tell the recovery team ahead of time because I knew they would freak out. So we told them right before, and this is my team in there getting to meet this baby, and it was such a surreal experience for all of them, and it made it that much more important. Now, when it comes to hospital development, early development is key. Early involvement is key. Meet with the staff ahead of time, explain the process and the role of the staff and each role that they may play. Share the birth plan so that adjustments can be made on their part. Um, some hospitals may want to run the process through their ethics committee. That's not uncommon, and it is not uncommon to meet resistance at first. Once you thoroughly explain the process to them, then they get on board and everything is smooth sailing. You just want to have all this done before mom goes into the hospital for delivery. And during the time that mom's in the hospital, make sure that you're present and you're there to keep the staff in the loop at all times, because that's vitally important to a positive outcome. Now, post recovery, again, what I typically do is I will leave the OR, and I apologize for my voice, y'all. Go back to mom's room and just verify, do you want baby back? Sometimes, if you've been in the OR for a couple hours, they've had that time of detachment that they may change their mind and not want to have the baby presented back to them. So that's something that you want to check on. And then, do they want the baby to stay in the room with them? Some hospitals have what they call a cuddle cut, and you want to do that. Make those arrangements with the nurse and the staff ahead of time as well. And then you also want to find out ahead of time if the hospital has a morgue or not, because that could determine, if mom doesn't want the baby back, then it's going to be on the OPO staff to determine where the baby will go. Whether it's going to be the funeral home, do we need to set up transportation? And also important is finding out if the family wants follow up after all this is said and done from the OPO. Most families are very involved with us, but some families don't want the follow up. The reminder is just a little too much for them. And as y'all know, everyone grieves differently. some of the keepsakes that we do for the family is a Build-A-Bear recorder. If you take the Build-A-Bear recorder and give it to the mom, she can bring it to her ultrasound appointment. They can record the baby's heartbeat. I suggest if you do this to have mom also record it on her cell phone so it doesn't get lost. She'll always have it. I had a family that their daughter was 2 and she was pregnant with her son who had anencephaly. And for Father's Day, they went to the Build-A-Bear. They brought the recorder, and she got to make her own bear. And Dad got to be involved, and it's something she carries the bear around all the time. It's super cute. We've also done hand molds, as you can see in this picture. That was from a local artist that volunteered her time to do that. We partnered with a little group of old ladies here. I hate to call them that, but that's what they are, that make burial gowns from old wedding dresses. So this is also something that we can offer to the families. And then, of course, the flag raising ceremony. And as Gina had mentioned earlier, the I Am star that they give for the families. Now, in addition to the keepsakes that we do, a lot of the families like to do their own. They may wear commemorative T-shirts. Some families have gone on to do their own fundraisers and dedications in their child's name. It may be that they put their child's picture on the hospital donor wall. We have a family currently right now that's doing a donation to have a cuddle cot donated to the hospital where her daughter was born. This is a picture of one of our donor moms. She handmade these hats herself and sent them to me, and they're for future babies with anencephaly because the hospital hats are so big. So these are very tiny and very fitting and just very special. We have had a few of our moms that were guests on our podcast, and we've had moms that accompany me to various presentations, not just around the state, around everywhere, really. These moms like to really get involved, and the one thing I hear a lot is, I just want people to say my baby's name. Say my baby's name, remember my baby. And so we try to do that as much as we can, every opportunity we get. Okay, again, this is Clark's mom and dad, and this was the flag raising ceremony that we had. And Clark's big sister, Olivia, wore this shirt. My brother's a hero. She wore it the day that they went into the hospital, and then she wore it the day that we did the flag raising ceremony. And you'll see a lot of families that wear these different kind of shirts. This is baby Austin. Austin also had anencephaly, and her mom is Kelsey. This picture right here is actually a smaller scale of a big poster that I have. And when I do outside presentations, I bring that with me. And there is a quote underneath it from Kelsey. I don't have that with me today. I should have put that on there. But Kelsey's very big into helping us out in the community. And she came to speak at, it's called an AWAN chapter meeting. I don't know if y'all are familiar with that, but it's neonatal nurses, all women's health nurses. And she came to help me present on neonatal donation and to tell her story. So our experience, y'all, I can't even begin to tell you. It's been unreal. I mentioned that these cases are unique. They're unique in the fact that we don't approach these families. All of these cases are family driven. They're doing their research. They're finding us, and they're full steam 100% into it. So anything that we can do, we do. We've been working really hard on getting education out to the public. A lot of these families, when they go to their physician doesn't know himself that donation is an option. So our goal is really to be able to work on getting handouts to these physicians, doing presentations, whatever we can just to get the word out there. The relationships that you develop with this family are unlike any other. Typically, these moms reach out to us early. So you're with these moms for a couple of months and these families for months, before the birth of their child. And so this relationship continues, and it's nothing for me to talk to some of these moms once a week. They'll call me or text me and just talk about everyday life. Community awareness has been amazing as well. Through doing this program, we've, like I mentioned, the photographer, but we've also found several different sources that we can use, sources that help with burial costs of infants. I mentioned the burial gowns, the photographers, any kind of monetary donations that these families may need. We recently just found a group that makes baby caskets. So that's something that we're going to reach out to and see if they can partner with us and what we can do. And really, these moments, they're just truly unforgettable. Each case, I walk away, I learn something new every time. And I can't imagine ever having to tell a mom, no, that this is not something we can offer for you. And when you have these moms, they're so selfless and so strong, and they just want their son or daughter to make a difference and to leave their own legacy. And when they call and ask if you can help with that, absolutely. I mean, like I put down here, it's the right thing to do. I did put my business card up here. So if anybody has any questions, y'all feel free to call me. I'll answer at any time, 24/7, weekends, holidays. I'm available. And thank y'all so much for letting me speak about this. And now I think it's going to be Miss Gloria Pryhuber.
Thanks very much, Laura. That's a hard presentation to follow, but I'll try to make it clear from a neonatologist and an investigator's point of view why these neonatal donations not only are important to the families, but are important to us. So I am here as representative of the LungMap Human Tissue Core. I also apologize for my scratchy throat. We have created the biorepository for investigation of neonatal diseases of the lung, or the Brindle Repository, as part of the LungMap program. And I just want to recognize my collaborators, Dr. Mariani Misra, Holden Wiltsey, Katzman, and Dr. Gail Deutsch, who's a pediatric pathologist at Seattle Children's, who are really responsible along with me for the work that I will show you. I'm also proud of my LungMap team members. These are our laboratory team members who are recognized by our Department of Pediatrics as Team Service awardees for the work that they have done for the LungMap project. What you're seeing there, maybe you're as mesmerized as I am by it, but the image that you're seeing is a video microscopy of lung tissue. Excuse me, lung tissue from a neonatal donor that is being imaged under a microscope. Multiple 2D levels are put together into this 3D video. And you're seeing the blue, the blue marks the nuclei of the cells, and the red is marking elastin fibers. So what you're seeing here are the mouth of alveoli being held open by those elastin fibers. You can see those come around into a circle and then go on out as you go through the layers. And you see the vasculature coming through, the vessels coming through that make this an organ that can exchange gas. This is a technique, actually, an imaging technique that has been relatively newly developed, and we wouldn't have done it without the neonatal tissues. The amount of information we have about these structures is actually relatively small, no pun intended, but rudimentary. And so this just gives you an idea of our ability to start to get at 3D lung reconstructions. Uh-oh. I think I need some help advancing the slides. Thank you. So as I said, I'm here as a representative of the Lung Molecular Atlas program. It was developed and funded by the NIH, the National Heart, Lung, and Blood Institute. And we are the human tissue procurement, or the human tissue core at the University of Rochester. And it was our responsibility to make available these young pediatric lung organs and tissues to the other research centers that are part of LungMap. So the Children's Hospital at Cincinnati, UAB, Yale, University of Pittsburgh, and University of California, San Diego. The Pacific National Northwest Laboratory in the state of Washington, and the Saban Institute at the Children's Hospital LA and University of Southern California. Our coordinating center has been at Duke and RTRI. Each one of these research centers has a particular area of expertise and evolving technologies that are allowing us to dig deeper and deeper into understanding how, in our case, how the lung grows, develops, and functions properly, and what goes wrong when it doesn't. Yeah, thank you. One more, please. So now I will give you just a little bit of background as to why I'm a neonatologist. Why am I so involved in this kind of study? This is a case story of a baby born with congenital lobar emphysema. Excuse me. This baby was born, oh, not quite yet. This baby was born at full term. So about 37 weeks, was known in utero to have a mass in the chest, in the left upper chest, and that was compressing the other parts of what should have been a normal lung. And we knew that this CLE might be a problem, and so the baby was intubated immediately at birth. And even with mechanical ventilation, you can see we were not able to get very much air into these lungs. They were hypoplastic or not fully developed and appeared immature. So within hours of birth, within an hour of birth, actually, that mass was resected, and you can see the air space where the mass was. A little bit of left lower lung that had developed, but not normally, and then the right lung that still wasn't expanding, was very stiff and difficult to ventilate. Excellent. We had increasing pneumothorax. There was leakage of air into the left chest, further compression of the lungs, such that the baby had respiratory failure and was placed on extracorporeal membrane oxygenation, where blood is taken out from a large vein, oxygenated in a machine, and then put back into the aorta to perfuse the body with oxygenated blood. You can maybe see that there's a chest tube on the right side now as well as the left. So this is an indication of how abnormally developed even the right lung was in this case. And we are stuck when we are faced with a baby like this. I have nothing that will make that lung develop faster or normally. And so this baby was on ECMO for 20 days and actually did quite well, was able to come off ECMO and at 1 month of life, still intubated and on high frequency ventilation, so on a ventilator that breathes at about 420 breaths per minute. And so this maintains life. It works with these lungs to be able to, for the baby to survive. But we do get complications. We get edema, we get fluid, and we get scarring of the lungs. By 2 months of age, this baby was still on that ventilator, but improving. The lung function was improving, but I think you can still see that there's abnormalities and scarring in those lungs. By 3 months of age, the baby was able to be extubated, so now only on oxygen. And then by 5 months, fortunately, was getting ready to go home. But I hope it makes sense to you that although the baby was able to survive, we have caused some injury to these lungs. There is some scarring, some abnormalities just from the oxygen and the ventilator exposure, as well as the developmental problem initially. So my frustration as a neonatologist is that I don't understand this process well enough to be able to intervene earlier, faster, better, what medications to give to avoid this chronic problem. And so I need to know more. And that is in part what our research is for. just to give you follow-up, the baby is now about 11 months old, is not requiring oxygen or a ventilator anymore, eats goldfish primarily, I'm told. Those are not the slimy kind. They are the cracker kind. The baby has oral aversion, which often happens. It doesn't want anything in his mouth. Often happens when you've been on a ventilator for such a long period of time. And nutrition is difficult for this baby. Is fed by a G2 route. And so again, I would like to be able to make our care of these babies better. And the best way I can do that is to understand their development and what I can do to improve it. Our preterm infants similarly are left with scarring in their lungs. This is a CT scan at 10 years of age of a baby that was born extremely preterm at about 23 weeks. And you can see that they continue to have scarring and cystic changes, abnormalities in their lungs in comparison to a full-term, now 10-year-old child. And these children's, the preterm children, not all, but some of them will go on to have emphysema and chronic obstructive pulmonary disease of adulthood. You can see even at 10 years of age how simplified their lung is in comparison to a full-term child's. So the Lung Molecular Atlas program was established in order to build an atlas of lung development on a cellular and molecular and organ level. This was to be dynamic and publicly accessible database, both with existing and generating new data. And we were to develop new tools, reagents, and technology to achieve molecular profiling of that developing lung. And overarching is what I just described. Our purpose really is to provide new knowledge upon which we can build better lung health and disease treatment, especially for children born too young, too small, and with problems of growth and development. So in order to do that, we had to think about what is it that it takes to make a lung, for a lung to develop. And just as with all tissues and organs, they are made up of living cells and of a variety of different types of cells, each with its own program, each with its own function, that's dependent on the DNA that is encoded in each cell, the transcription of that DNA into RNA, and then that translation of RNA into proteins and lipids, the combining of those proteins and lipids to then become that individual cell with its own particular function. These cells come together in community, and then those communities form structures. And for the heart, I'm sorry, yes, for the heart too, but for the lung, those structures that are particularly important are the blood vessels, the airways, the gas exchanging membranes, and the immune system. And it might be surprising to you actually to realize, again, how rudimentary our understanding is of these structures and what makes them form properly and how they respond to injury, make them respond better and to repair. So our goals for LungMap were to comprehensively characterize the diverse cell types of the developing human lung. We don't even know actually how many cell types there are in the normal lung. Just recently, a researcher described the ionocytes, which had never been described before. And then to understand how those individual cell types function and how do they function together. This image is just to give you, again, a better sense of what I mean by how many cells and how they function together, work together. This is an airway that has cartilage. It has smooth muscle cells. It has glands that secrete mucus and thinning fluid that keeps our airways hydrated and clear of organisms and clear of environmental bits that might end up down our airway. There are multiple different types of cells just in this epithelium to support that structure. Then, of course, we have the alveolar structure where the gas exchange occurs, and there are multiple different specific cell types there, including the blood vessels that bring blood close to the air to exchange gas. It is remarkable how these need to function together to make a normal lung, all of these many cells. This particular area is also important in asthma. Not only is the work we do important for newborns, but also for people who live as children or adults with lung disease. Also for the lung map, we needed to represent normal lung development from what's called the late cannulicular stage through alveologenesis up to young adulthood. This cannulicular stage, excuse me, is the youngest point at which we currently have human viability, so about 22 to 23 weeks. And prior to that, the lung develops as a branching airway system, and we have a fair amount of research and data about that from abortuses, from early fetus. But because of a lack of access to neonatal tissue and young child, we have had relatively less information available about what is normal lung development through these early adult years. So when lungs come to us, this slide just diagrams that we have a process. It's a standardized process. We ask for the lungs to be en bloc, and each portion of the donor material that you send to us is designated for a particular purpose. I mostly want to reassure you here that nothing is wasted and nothing is discarded. All of the tissues are tracked and documented in our repository inventory, and that my lab staff and all of the investigators that I work with realize how important these gifts are to their families. These are highly prized to us and respected. To give you a little bit better sense of what we actually do, I'll walk you through this slide. And so when the lungs come into us en bloc, we intubate them and inflate them with air. And then we're able to do high resolution CT scans. These are higher resolution than you could get in a living patient. But it gives us a chance to look especially at these outer areas of the lung and what those structures look like. We are able to count dividing airways, determine length and size. We have now computer savvy investigators who are developing common coordinate frameworks on which we can put all of the additional data and figure out where things should be in this normal lung. I also wanted to point this out to you. This is one of these neonatal lungs that you have sent to us. And they really are quite beautiful. They are pink, they're light, they're like marshmallows when they're inflated with air. When you see them, they may look rather bruised, rather bloody. But if you can give us even a brief flush, and then we can inflate them, we can demonstrate how beautiful and normal they really are. We then take those lungs, and each lobe, as I said, has its own purpose. The lower lobes we inflation fix. Some go in paraffin, the other gets cryopreserved and frozen. The left upper lobe is inflated with a freezing material and then divided and frozen, and then we dissociate cells from the other remaining right lobes. Everything we do is photographed, and that's in part just for our record, but it's also so that we can track back to a particular part, particular position in the lung, any of the further analysis that we do. So for example, this lobe was standardly sliced. One of those slices then is shown here, made into blocks, and each block has an address of where it came from in that lung. This would be that C2 block. We then can section those blocks, and they can be used for standard stains, H&E stain shown here, and also for immunohistochemistry and in situ hybridization to look for proteins and RNAs. And technology continues to advance here. We're able to do multiplexed. We're able to look at many RNAs and many proteins in the same section of tissue, which has advanced our knowledge of how these things work together and how the cells are arranged. In the cells that we dissociate, I can show you here that the mean viability of those cells is about 95%. So these lungs, although they are post-mortem, the cells remain alive for a prolonged period of time. We can take those cells, and this shows you using flow cytometry to sort them into mixed immune cells, endothelial cells, epithelial cells, and remaining mesenchymal or structural cells. We can do further sorting down even to single cell capture of these cells. We can take those sorted cells, we have taken those sorted cells and looked at them for their particular RNA gene signatures. So each column here is one of those cell types. Each very thin row is a gene. Red is highly expressed, green is very lowly expressed. And you can see that the mixed immune cells have certain genes that they express. The mesenchymal cells, the epithelial cells, and the endothelial cells can be well distinguished by their gene expression. And this only shows you the genes that we know to be markers of these particular cells. In doing this, we're able to identify other genes that we didn't know might be specific to a cell, or that maybe is not specific. It helps to explain the differences between these cells and how they change over the lifetime, how they change with development. We've been able to do something similar with the protein signatures. So PNNL is able to take these sorted cells and look at proteins that are highly expressed in each cell type. Again, things that we already knew, and then identifying proteins that we didn't know existed there. And then similar sorts of things can be done with the lipids. Taking it down to a single cell level, we're also able to make maps of these single cells. This happens to be based on what the DNA accessibility looks like. And each cell is represented by a dot, and as you can see, certain dots, certain cells cluster together because they're similar and different from a different cluster. And you can also see here that the map changes with age between 1 day and 9 years old. So this is just changes in the DNA that allows it to be made into RNA and then into protein. So lots of information is coming from each one of these cases. So what have we accomplished? We've collected over 200 cases ranging from 30 weeks to 10 years of age. By my count, about 75% of them have actually come from IAM, and we're very grateful for that. Our age at birth, 40 of these cases were born preterm. Age at donation ranges 70 newborn, 44 infants, and 100 children. 83 of these 200 cases, or over 200 cases, are deemed by the pediatric pathologist to be normal in growth and structure. And those are particularly important to us because we can't study disease, we can't understand disease if we can't understand normal. But we've also been able to add to the repository several known cases of lung disease, asthma, bronchopulmonary dysplasia, the disease of prematurity, and pulmonary hypoplasia, the problem of the lung not growing appropriately. We've been able to provide these cells and tissues along with important information that you can provide us, clinical information, a review by our pediatric pathologist, provided them out to the research centers and to the broader research community. The Brindle, the lung map, is not meant to be just for the lung map investigators, but these cells and tissues are available to investigators around the world that qualify, of course, as investigators in their area of research. We've demonstrated critically that these human cells and tissues are of very high quality. There were a lot of naysayers who thought that because the donor was no longer alive, that the material would not be instructive to us, that there would be too much artifact. But we have indeed demonstrated the quality and what we can learn, and that our new technologies, some of which didn't even exist when we started, how those technologies can be applied to these tissues. We've generated multi-scale, high-quality molecular and imaging data, excuse me, and developed websites, the Children's Hospital web portal at Cincinnati, and the LungMap website. Just to highlight some of what's been added, we've added first of kind, it's never been seen before, single-cell RNA transcriptomes of the developing lung. This will help us to identify those novel cells and their functions. Excuse me. We've also been able to identify lipids and proteins, novel markers, developing new tools, and sharing the imaging. I can tell you that we've only begun. Maybe one day we'll learn to treat the respiratory syncytial virus that I have that's causing this cough and cold. But truly, we are at the tip of what we learn and what we will be able to do. And then I'll just leave you with a few thoughts about the importance, particularly of neonatal donation for research. Our ability to study neonatal human tissue and cells is essential for the research that unequivocally saves lives, improving diagnosis and treatment. Without this human tissue research, we are stuck learning from animal models. And it should be very clear that mice are not human and children are not small adults. The mouse immune system is entirely different from a human's. Not entirely, but very different. And as is their airway, they are missing actually portions of airway that are present in human. With human tissue research, we can work forward on personalized medicine, gene discovery, diagnosis discovery, network discovery. How do these genes, proteins, and cells work together in an organ? Drug discovery and testing. It's been very frequent that drugs developed in animal models, tested in mice, actually don't turn out to work in human. There is just no substitute for working with the human materials directly. With human tissue research, we can figure out what is normal, what's normal variation, what's abnormal, what needs to be treated. And then we're coming to understand that adult disease, many of them start in infancy and even in fetal life. So that neonatal research is particularly important to try to understand what's underlying those adult diseases. As we go on to identify these genes and proteins that are necessary for lung growth and function in the normal patient, we will find diagnoses for diseases that we haven't understood previously. We'll be able to describe that abnormality, define the diagnoses, determine prognosis, and find ways to reduce the disease. And I can assure you that it will not be just one child who's saved by these donations of gifts for research, but many, and in ways that we can only just imagine now as our technology and our understanding grows. These gifts will long outlive their recipients, and I do want them to be my legacy. Just a recognition of all of the people that have contributed and have done all the work that I've explained to you, both at the University of Rochester in our cores, or imaging sciences. Also, our collaborator in Seattle. I can't thank IIAM and GINA enough, our other research recovery organizations, and especially those of you on the phone who are organ procurement people. I know that this is a difficult job to approach and work with neonatal cases, but I hope I've convinced you how important it is. And then, of course, I would want to thank our donating families for their gifts. And I thank you for listening.
Thank you, and thank you all for a truly incredible, informative presentation. If anyone does have additional questions, you can enter them in the question section now. I did want to start with one for Laura. So, considering that the majority of our attendees with AATB are on the OPO or tissue recovery side of things, who commonly completes the recovery for you all in neonate cases?
In the neonate cases, we have our lead, like our team lead position would be our research manager. And then usually we'll, if we can take someone from the tissue team or a scrub tech, we kind of put it together when these cases happen. But it's usually, it would kind of be like a tissue recovery.
Hmm. And Gina, that we talked about completing the DRE prior to these cases. In review of a medical history, what is something that would preclude a family from having the opportunity for neonate donation?
There's, if there was an indication of a positive serology, and infection might be something where the researcher may have concern of risk. But outside of that, and I might even defer to Gloria, I think that the researchers are incredibly liberal on what they will accept with these donors when it comes to, again, a family history, because they have really never worked with this type of tissue. So they're willing to basically blindly receive these gifts and see what they can learn from it. Gloria, is there anything else that you would talk about as far as the family history, not so much the baby?
Right. So as you said, the high level infection that might put us at risk, we would decline. But there really isn't very much else in the family history that would, for me anyway, as a investigator, that I would turn down. I think what is most important actually is that we know the history. So the history of the family, if there's chronic disease that runs in that family or something in the mom that might have affected how her baby developed, that's important history for us so that we can interpret our data correctly. And then similarly about the baby, to understand what maybe there was nothing anomalous about the lung that we know of, but were there other organs involved? Was it just anencephaly, or were there other organs? We also actually, how much amniotic fluid there is informs us about lung development, because you're dependent on that amniotic fluid, and you're actually dependent on breathing it and swallowing it for the lung to develop normally. So that's a piece of information: gestational age and amniotic fluid volume, and anything else that was known by ultrasound before that baby was born is really important to us.
And there might also be circumstances where there's evidence of sepsis, or obviously we talked about infection, and I think that tends to fall into the bucket of serological infection. But if there's any other type of, again, broad infection, meningitis, that would, I think, also be a deterrent.
Yeah, I would agree. It depends on the funding too. Actually, LungMap was supposed to be normal. We were supposed to archive just normal cases. But it's very clear, again, it's important to have normal and to be able to really do the job we want to do. So we will often take disease as well.
So as we run to the end of our time, I think one thing that would be important to highlight, and this would be for Dr. Prahuber, and for Gina, what resources are available that the donation community and transplant communities can use to inform hospitals, their colleagues, about the work that's done through these great donations?
Mm-hmm.
I think at this point, I'm going to kind of point to the community at large. Anencephaly is such a unique diagnosis, but it is a growing community that organically and in a grassroots manner are together building resources. So that tends to be the target audience. As you saw before, 60% of our donors have that diagnosis of anencephaly. So that community is pushing information, for others who are getting a diagnosis that is a non-survivable neural tube defect. So that's what we have heard so much about, is that how are hospitals or families, and therefore the OPO or tissue bank, how are they able to give or point a family to a direction that would help them? And a lot of it is those families that have already walked down this path. IIAM has a lot of information on our own website, but we also have a gigantic list of other sources that are on that website that will point to other sources of materials and information for anyone interested.
Great, thank you so much. And there are a couple additional questions, but we've ran out of time. If anyone does have follow-up questions, please feel free to enter them in the survey in the post-program evaluation that'll come out in an email in just a little while. And to Gina, Laura, Dr. Pryhuber, thank you so much for taking the time to present this, and it's much appreciated.
You're welcome. I'd be happy to talk to anyone who would like further information.
Great. Thank you all, and have a great day.